Hello, in this video we're going to view the Nobel Prize in Physiology or Medicine for the year 1906. We're going to look at the Nobel Prize lecture, or better a double lecture, one by Golgi and one by Cajal on the nervous system. We start with the Nobel Prize lecture of Golgi, the Neuron Doctrine, Theory and Facts. We can say in advance that Golgi was not in favor of the Neuron Doctrine. He had his own theory, more of a network theory. And he was polemizing with Cajal, who was following the Neuron Doctrine. In the end, history is more favoring the Neuron Doctrine of Cajal than the network theory of Golgi. But in the year 1906, the matter was not settled. It was only later. Okay, we're going to start with the summary of the Golgi Nobel Prize lecture, summarized by me, of course. Golgi had long opposed the neuron theory when it is in general going out of favor, but the majority of the involved scientists still support it. Well, that has been disproved. The word neuron tends to be misused, and many authors substitute it by nerve cell. But everyone agrees there is a continuity between the cell and the nerve fibers. Since 10 years, the idea that nerve cells form an anatomical unit becomes acceptable. Waldmeyer perceived the existence of such a unit and called it the neuron in 1891. He wrote, "The nervous system is made up of innumerable nerve units, called neurons, which are anatomically and genetically independent of each other. Each nerve unit is composed of three parts: the body, the fiber, and the terminal branches." End of quote. Motor impulse travels from the body to the nerve terminals. Sensory impulses go the other way. Later it was better defined. One neuron is an embryological unit, two, the neuron is one cell, and three, the neuron is a physiological unit. Best expression of the neuron theory is the theory of dynamic polarization by Van Geheugden. See the Cajal lecture. The transmission of the nerve impulses is from the cell body to the nerve extensions, not the other way around. Each nerve has a receiving and a conducting apparatus and a transmitting organ. Protoplasmic processes conduct towards the cell body, nerve process away from it. Golgi found that the pathway from the periphery goes directly into the spinal cord, avoiding cells. Golgi explained that on details he does not agree with Cajal, and details skipped here. If you want to read more about the polemizing, use the original lecture. Figure 1 and 2 show the formation of the nerve network. We will see this illustration soon. Back to the neuron theory. Now, Golgi disagreed with it, attaching more importance to the network, which he calls a nerve organ. Every element of the central nervous system contributes to the formation of that network. Later, Golgi will return to the protoplasmic processes, now further with the neuron theory. 3 points. 1. The neuron is an embryological unit, thus derived from a single cell, as described by Waldmeyer. The nerve fibers are direct emanations of the neuroblast, and the independence of the neuroblast is proven using methods with ordinary staining methods, using Golgi's new black staining method. One can follow the development better. Golgi believes that an intimate connection between the parts exists from the start, but it's hard to prove. In analogy, cells in the Malpighian layers were found to be independent, but are now proven to be connected with fibrils. Are the connections there from the start, or do they develop later? It is difficult to answer. Some research supports the one and some the other idea, so it wasn't settled at that point. Golgi takes the position that neither position is proven. Golgi disproves of the idea of both yours and Besta and proof of their theories. I skipped the details, but if you want to read it, read original, quite lengthy lecture. Then 2. The neuron is, in the adult state 2, an independent cell. The research with silver nitrate preparation have led to the understandable conclusion of cellular unity. But Golgi states that there are two types of nerve cells, not one. Type 1 and type 2. respectively lose their individuality in the nerve processes. See figure 3 and 4, who show type 1, and figure 5 says who show type 2. Golgi underlines that the two types behave in a different way, but are both found mixed in the same regions of the nervous systems. Here you see some of the illustrations. That was just a pointer. Here you have the type 1, for instance. It will be more easy to distinguish between the function of the two types if they were found in separate places. The spinal cord was found to best to investigate this, but even there it is no longer the case that motor and sensory functions are completely separated. It was believed for a time that this was so. They are connected with fibers. Golgi was able to prove that in the spinal cord sensory and motor fibers behave in opposite way. Motor fibers, direct and detached contact with the ganglion cells. Sensory fibers, indirect contact through the diffuse network. The second type loses individuality in the loose diffuse network. Golgi recognized the existence of this network even before the rise of neuron theory as a true organ found in the layers of the gray matter in the central nervous system. Its structures seem to be of great importance in relation to its functions. Elements are 1. Collateral fibrils, which radiate from the nerve process of cells of type 1, and 2. The assemblage of nerve processes of type 2, breaking up in a highly complicated way, and 3. Collateral fibrils coming from nerve fibers of type 1 to make direct contact with ganglion cells of type 1, and 4. A large number of other nerve fibers which, like the nervous process of type 2, gradually lose their individuality while dividing to become extremely thin filaments. Golgi has demonstrated the existence of the network in the spinal cord, the cerebellum, and in the cerebral cortex, a little different in each. See figures 7 and 8 for the network in the cerebellum, respectively the hippocampus. You can see that shortly. The existence of a network with functions does not mean that there is fusion of one fiber to another. Fibers do not always have an insulating myelin sheath. When they run side by side, communication is possible. Thus, communication without direct contact is possible. Therefore, Golgi does not accept the idea of independence of each nerve cell, the basis of the neuron theory. He points to the organization of the dentate gyrus, which illustrates the group action of nerve cells, thus opposite to the alleged individual actions. Figure 9, here you see those figures, which was referred to earlier. Then the diffuse network is exclusively formed by filaments that are derived from both types and from fibers. This network is a liaison between different parts of the nervous system. Nansen earlier also concluded that the true organs of nervous activity is the fibular network rather than the ganglion cells. Petter agrees, but until now it cannot be proven. Until now is 1906, of course. The protoplasmic processes, according to the theory of dynamic polarization, are also important. Golgi rejects a careless idea and the constant presence of anastomosis. The processes are subdivided with fiber endings contacting both vessels. See figure 1, maybe also for nutrition. Parts of the brain, like the corpus quadrigeminum, are without protoplasmic processes, are to compensate and closed by capillaries. Calcic degeneration seems to begin in the smallest bunsen of the protoplasmic processes and then progresses centrally. It is well possible that the protoplasmic processes share in the function of the nerve cell. The pericellular and peridendritic network must also be mentioned here. It is so-called Golgi nets, but it's not the same as the diffuse network Golgi spoke about. Then Golgi returns to the sheath, which is not well understood, but sometimes to appear to be reticular. Also, problems of the structure of the nerve cells is far from its solution. Modern researchers focus on cell structures and thus serve to give arguments for or against the neuron theory. Golgi rejects the ideas of Nissel because they are too speculative. He also rejects Better's ideas, who seem to understand what Golgi means with the diffuse network. Golgi approves of the methods of Cajal, but it does not always lead to converging results. It is thus not the time to end the debate on the neuron theory. Golgi approves of the work of apathy, but it is one in vertebrates and cannot be applied to vertebrates. Golgi then shifts to the studies on the ancillary rectangular apparatus, see figures 11 to 14, which is also an unresolved problem. The same goes for the superficial nerves we can see in figures 15 and 16. We can only say that they are different from the internal reticular apparatus. Here you see those figures 10 to 14. Golgi points to the work of Holmgren and reticular apparati, but again there are differing facts. Betty and Donaggio both showed the exquisite fibrillar structure of nerve cells, but Betty emphasizes the independence of the fibrils, while Donaggio takes the opposite position, underlying the complex reticular arrangement. Figure 17 is taken from Betty and figure 18 from Donaggio. Golgi seems to take position in favor of Donaggio in his neutral to Cajal and against Betty and Bishovsky. Cajal's work on the structure of the nerve cell is praised. See figure 19. Cajal is probably how it's said, showed the fibrillar structure in detail even at the beginning of embryonic life, but still is not clear on what fibrils do. There are still conflicting results. Then 3. The neuron is a physiologically independent unit. Wallace's law is at the origin of the doctrine of physiological independence of the neuron, but for Golgi, this proof is worthless. The same for the proof that systematic degeneration of nerve cells. The doctrine of dynamic polarization is not the only possible for him. The idea of cerebral localization had to undergo profound modifications. The localization of functions is imprecise, and substitution and compensation is possible. It is clear that a single nerve fiber may have connection with an infinite number of nerve cells, many far away. Nerve fibers from the periphery are more connected to the central nervous system than with their own surroundings. Then a quote, as for the specific function of the central nervous system, I have on several occasions contested that it was correlated with a specificity of organization of the nerve cells, and I have come round to the idea that specific function is not associated with the characteristics of the organization of centers, but rather with the specificity of peripheral organs, destined to receive and transmit impulses, or again with particular organization of peripheral organ which must receive the central stimuli. Golgi then repeats that there is no reason to accept the independence of each nerve element, giving the network way of functioning of the nervous system. Here more of the illustrations referred to. The same for the opposition action of the sensory function. One cannot dismiss that unitary action of the nervous system, concluding, a quote again, when I think how the modern period of scientific research, whose developments I have had the good fortune to witness, has really enlarged in a surprising way our knowledge of the organization of the nervous system, my wish is that these new anatomical studies may represent a new element of progress for humanity. This is, of course, a summary of the lecture. Not all arguments could be taken up. Otherwise it would have been too long, but if you're interested and you want to read his complete argumentation, follow the link which will also be in the description under the video. Again, I want to underline that this is a lecture which is summarized quite a bit. In general, Nobel lecture is about 10 pages in a PDF. This is about 3/4 to an hour of speaking time, but both the lectures of Golgi and of Cajal were about at least 3 times longer. So this makes it difficult to summarize a lecture and maintain a logical argumentation. Yeah, so that's why it's probably a bit sketchy, and which is also will be the same for the second lecture given by Santiago, Ramón y Cajal on the structure of connections and neurons. He was, of course, a defender of the neuron theory against Golgi, was a defender of the network theory. With hindsight, Cajal's neuron theory has become dominant, but that was only later after the investigations into neurons and The central nervous system was ameliorated by the use of the electron microscope. That was not in 1906, but much later. Okay, let's go to the Cajal lecture. Here you see some of his photographs he made, coming from also from the Nobel Prize side, of course. On the article, there are 3 articles on this subject, one on Cajal, one on Golgi, and one on both of them. And these are some of the illustrations from it. Okay, let's go to the lecture itself. Summarized by me, of course. Again, it was a long lecture, so it has to be summarized a lot. So sometimes the argumentation may be a bit sketchy, but if you're interested, you can always read the original lecture. The link will be in the description. First, some general conceptions. 1. The nerve cells are morphological entities. Neurons. This was first stated by Waldmeyer. We already saw that in the previous lecture. Little was known about the axon branches and collaterals. Using Golgi's coloring, we studied the central nervous system. We is, of course, Cajal and his collaborators. The ramified fibers always proceed to the neural body or protoplasmic expansions. The nerve elements poses reciprocal relationships. The contacts created perpendicular systems, the cell bodies and dendrites conducting like the nerve fibers and the axons. Protoplasmic expansion also play a conducting role towards the cells in axons away in dendrites. Dynamic tubularization, Van Gogh and Cajal. No research goes against the above. Now striking examples of the interneural connection. The single prolongation of the sensory corpuscles does not penetrate the gray matter. There's an ascending and a descending branch. See figure 1. We'll see that shortly. They end finally in the gray matter. The sensory fibers exist in 2 varieties, long or reflexomotor, destined to contact a motor neuron, and short and destined to come into contact with phenolic neutrons. Figure 2 shows 2 important currents, a direct reflexomotor current which transmits with going via a neuron, a indirect association reflectomotor current which takes a detour via a neuron with actions in the white matter, so 2 ways. Figure 3 shows the connection of visual fibers and the cells of the retina. The retina can be considered as a ganglion with 3 rows of neurons, all 3 interconnected in plexiform layers. The internal plexiform layers are divided into layers again with complex connections. The axons of the neurons we find in the mid and the intermediary brain. A third and a fourth connection are recently found. A further argumentation I have skipped. See figure 7. Figures 4 and 5, who neurons, connection cells and fibers in the cerebral lemma, again 3 layers of neurons. All the neurons have 2 sources of connection. Intrinsic connections between the neurons of the layers and extrinsic connections with the cerebellum and other central organs. Cajal deals with both here. Cajal underlines that both connections are also interesting theoretically, underlying the truth of the neuronal doctrine. Talked about that. Here you see some of the illustrations referred to. If you want to study them more deeply, probably you go to the lecture itself, this link. Most fibers are large medullary tubes which ramify and end in the granular layer in contact with the digitiform branches of the grain cells by means of their rosettes. The fine growth of the moss fibers form a loose neurofibular rectilium. As regards the climbing fibers, they cross the granular layer and develop ascending trunks and branches. The result is that the gram cells and the Purkinje neurons can receive nerve impulses from other centers. Then to the centrifugal fibers, shown by Cajal and Doctrine, are found in the retina and other regions in the brain. See figure 6, which shows the retina of a bird, and figure 7, an example of the centrifugal fibers of central organs. Cajal deals with the structure and workings. Again, skip here. Only mention here the role of the centrifugal fibers. At the moment, we do not know. In the meantime, the research into the morphology of the refigurations of neurons is finished. The neurologists now look at the constitution of nerve protoplasm. Nissel discovered the basic phylic bodies of the protoplasm, which was a fortunate start. Apathy and Beatty continued research, but it was difficult. It was led to the neurofibrillar procedure of Sumaro, Cajal, Wilczewski, Donaggio, Ligaro, etc. Cajal reduced silver nitrate method gives good results in some cases. The same is true for other methods. The following additions were made. A. The neurofibular framework of the neutrons of vertebrate is composed and of many independent conductors, but is a continuous network with different kinds of connections. See figure 8. B. The same reticular disposition can be seen in the variocosities and enlargement of the nerve branches in the motor end plates, the sensory endings, the outgrowth of the most fibers, the tip of nerve fibers that are regenerating. See figure 9. The idea of Beatty and apathy that these filaments are the sole conducting organs of nerve protoplasm cannot be accepted. See, the neurofibrillator reticulum is not a fixed, stable conducting system, but a framework capable of undergoing notable changes depending on the physiological stage and on the pathological conditions. Here again, some of the illustrations referred to. Figure 10 to 12 shows several of these changes in the protoplasmic network. Figure 13 shows the transformation of neurofibrils on the central end of the crus nerve, with the neurofibrils regenerating. This shows that the neurofibrils have a certain autonomy and are capable of change, forming structures of variety and variability. Recent research with the reduced silver method has revealed something of the morphological order. A. The existence in the neurons of the human sympathetic chain of remarkable dendrites only found in subcapsular space, figure 13. The terminal ramifications of these are extraordinarily complicated. See figure 15. And then B, the presence in the same sympathetic ganglia in men of glomeruli or connection plates. The discovery in the sensory ganglia of the cells with the protoplasm is partially disposed in a system of window cells. See figure 16. The presence of special appendices ending in encapsulated balls in certain neurons. See figure 17. And then E, the existence during regeneration of spheres possessing a neurofibular network. Again, some of the illustrations referred to. All this has made the neurological doctrine of His and for all acceptable by many, but in reality it cannot yet be accepted as an unimpeachable dogma. Maybe in the future new correlation processes can reveal how new connections between neurons. It would be very convenient if all nerve centers were made up of a continuous intermediary network between the motor and the sensory nerves. But we do not know, but there's no reason for skepticism. We need to continue the work and wait for what the future brings. Several physiologists and zoologists have adopted the doctrine of propagation of nerve currents by contrast or at a distance, but their allegations are based on incomplete methods. In the end, neurological conceptions show more results. Morphological objections to the propagation of nerve endings, Cajal and other neurologists have searched but not found the supposed phenomena. The histological arguments of the opponents of neurological doctrine show only lack of foundations for the hypothesis. Here again, some of the illustrations referred to. If you want to study them, best download the PDF from the Nobel Prize site. Then the final quarter of the long and tedious lecture, and long and tedious are Cajal's own words, contain the following 3 chapters or paragraphs: proof of the neurogenic doctrine of Kupfer and His, proof derived from the regenerative mechanisms of nerves, and proof derived from embryonic neurogenesis. In this chapter, Cajal polemizes with his opponent and criticizes the competing theories. This part of the lecture is already quite condensed and thus difficult to summarize, and therefore I have skipped it, except for the conclusion at the very end. More of the illustrations, which is it. To sum up, the entirety of the observations which we have just shown, and for many others about we have not the time to talk, the doctrine of neurogenesis of His is clearly revealed as an inevitable postulate. Ramón, the scientist who in the last years of his life so well filled, suffered the injustice of seeing a phalanx of young experimenters treat his most elegant and original discoveries as error. I finish by greeting most warmly and cordially this alert and sympathetic assembly, which I ought to thank much for their attention and kindness to me during such a long and tedious lecture. Again, it are his own words. Well, as I said, both lectures are very long, and I had set myself to summarize them within an acceptable time limit of about 25 minutes. When you read the complete lectures, you get it's about 3 hours each. So therefore, as I said, the argumentation may be a little bit sketchy. Maybe it is better to, when you want to know more about it, to read information on the Nobel Prize site first. For instance, the award ceremony speech, which explains why the Nobel Prize was given to these. This is also in the previous video. But there are also 3 articles, which I have already mentioned. One on Camillo Golgi, one on Santiago, Ramón y Cajal, and one on them both. And it's probably better to read these articles first. And if you're still interested and you want to read more, then go on to the lecture. Well, for the moment, thank you for watching, and maybe till next time when we will go to the 1907 prize.